So for chapter 7, we are going to talk a little bit about energy flow, oxidation and reduction reactions, coenzymes, the mitochondria, and the different steps that we do to make ATP, and the difference between aerobic and anaerobic cellular respiration and fermentation. This heading from this slide came from a textbook, and it says life is work and requires energy, which I think is a little bit funnier than it should be, but it's a true statement. So in most systems, energy flows into the system as sunlight and out of the system as heat, and photosynthesis is used to generate carbohydrates and oxygen. With that carbohydrate, that chemical energy is used to generate ATP, which powers work, and ATP can be made through cellular respiration, and both photoautotrophs like plants and heterotrophs like animals rely on the carbohydrates generated by photosynthesis. So cellular respiration and photosynthesis both involve oxidation reduction reactions or redox reactions. Oxidation is the loss of electrons and reduction is the gain of electrons. And for most biological redox reactions, we can track an entire hydrogen atom with its electron, which will be either removed or gained. In this image below, we've got reduced compound A. It's reduced because it has those electrons. And then you can see this reduced compound A can give its electrons to what was oxidized compound B, and then B becomes reduced compound B, and since A has lost its electrons, it becomes oxidized compound A. So a couple memory tricks. In chemistry, I think they say oxidation is loss of electrons, reduction is gain of electrons, so oil rig is one mnemonic or memory trick. I like Leo the lion says grr because it tells you what you're losing. So loss of electrons is oxidation, Leo, and then gain of electrons is reduction. We talked about coenzymes in a previous chapter. They work together with enzymes, and in this unit we'll focus on coenzymes as electron carriers. Reduced coenzymes are when the coenzyme has been reduced and picked up those electrons. In cellular respiration, NAD picks up hydrogens and electrons and becomes NADH. FAD becomes FADH2 or FADH plus H. In photosynthesis, NADP is going to be the coenzyme and it becomes reduced to NADPH. So in this picture below, this first thing is not a weird googly-eyed monster. It's an organic molecule with two hydrogen atoms attached to it. And so when NAD, our coenzyme, picks up those hydrogens and electrons, it becomes NADH plus H, which is that reduced coenzyme. And then that organic molecule, since it lost those hydrogens and electrons, it is said to be oxidized. We talked about this before as well, but there's two big ways we can produce ATP. We can do oxidative phosphorylation, which produces the bulk of ATP. It uses an electron transport chain and a proton gradient and chemiosmosis. So there's this protein pump in the inner mitochondrial membrane, and there's these hydrogen ions, and there's a concentration gradient, and the hydrogen ions flow through ATP synthase, and that provides the energy needed to produce ATP. There's also substrate level phosphorylation. Substrate level phosphorylation produces less ATP. It's used in glycolysis and the citric acid cycle. And in substrate level phosphorylation, an enzyme transfers a phosphate group directly from a substrate molecule to AD. So that brings us to cellular respiration. Cellular respiration is not the same thing as breathing. I think sometimes people get confused about that. Ultimately, though, cellular respiration is the reason that you do breathe, because cellular respiration requires oxygen and releases carbon dioxide. Technically, some organisms don't need oxygen, but we'll get to that later in this chapter. So what cellular respiration is when we break down organic molecules like glucose and release energy, and that energy is used to produce ATP. This is a redox reaction where we have glucose and oxygen going in, and we get carbon dioxide, water, and energy used to make ATP going out of that overall reaction. So let's do a couple review questions on oxidation reduction reactions. So in our first reaction, we have 4 NADH plus O2, and we get 4 NAD plus 4 H2O. So we want to know what's oxidized and what's reduced. Pause if you need to. So the NADH is oxidized to NAD because what's oxidation again? Oxidation is loss of electrons. So NADH loses its hydrogen and electron and is oxidized to NAD. So that means that oxygen, the O2, picks up hydrogens and electrons, so oxygen is reduced to water. For number 2, we've got glucose plus NAD, and we get pyruvic acid and NADH. So again, pause if you need to, but what is oxidized? So the glucose is oxidized to pyruvic acid because, again, oxidation is loss of electrons. And then the NAD is reduced to NADH. In question number 3, we've got acetyl coenzyme A plus FAD plus NAD, and we get carbon dioxide, FADH, and NADH. What's oxidized? Acetyl coenzyme A is going to be oxidized to carbon dioxide, and then the FAD is reduced to FADH, and the NAD is reduced to NADH. So then what's the main point of cellular respiration? The main point of cellular respiration is ATP production. So there's 4 main phases of cellular respiration. This is a really good preview slide because it gives us the 4 main phases and it tells us where it's going to happen and kind of the big picture of what's going on. So cellular respiration can be broken down into glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. Honestly, we use multiple molecules besides just glucose, but we're going to focus on glucose because we have to start learning part of the process somewhere. And then in this picture, this thing that looks like this jelly bean down here, that's the mitochondria. So this picture is showing us that glycolysis happens outside of the mitochondria, but the other 3 reactions happen inside the mitochondria. So we need to talk about mitochondrial anatomy in order to understand this process a little bit better. So the mitochondria has an inner membrane, which is called the cristae. That's the location of the electron transport chain. And then the mitochondria also has a fluid portion, which is called the matrix. And that is the location of the PrEP reaction and the citric acid cycle. So that brings us to glycolysis, which is that first reaction in cellular respiration. And for all of these reactions, we're going to focus on the location, reactants, and products of the reaction. So glycolysis does not happen in the mitochondria. Instead, it happens in the cytoplasm. Glycolysis needs glucose going in. It also needs NAD and ATP, and it produces 2-pyruvate, which is a 3-carbon molecule, 2-NADH, which is a reduced coenzyme, and you get a net gain of 2 ATP per glucose molecule that goes through glycolysis. Glycolysis is divided into the energy investment phase and the energy payoff phase. Don't get super hung up on all of the steps, but sometimes the steps in the process helps make the rest of the process make sense. So this is still glycolysis, but we're starting with a 6-carbon molecule glucose. We're spending 2 ATPs to basically stick phosphate groups on either end of that glucose, and then we split that 6-carbon molecule into 2 3-carbon molecules. Then in the next part of glycolysis, basically what we're doing is pulling those hydrogens off of those 3-carbon molecules, so we get NADH, and we also have the energy to produce 4 ATP. However, we usually don't say that you produce 4 ATP in glycolysis. We typically talk about the net gain of ATP being a net gain of 2. So let's review. Where does glycolysis occur? Glycolysis happens in the cytoplasm. Number 6, does glycolysis require oxygen? No, glycolysis does not require oxygen. And number 7, what is the name of the 3-carbon molecules at the end of glycolysis? That would be pyruvate. So we made those pyruvates or pyruvic acid. So what happens with them? Well, that really depends on whether oxygen is available or not. If oxygen is available, we go on to the oxidation of pyruvate, which is the next step, then the citric acid cycle, then the electron transport chain, which our book calls oxidative phosphorylation and chemiosmosis. So it continues on with something called aerobic cellular respiration. That root word aero means air, and in this case we're talking specifically about O2. If oxygen is not available, then the cell is going to switch to either fermentation, or if it's capable, it will do anaerobic respiration. That root word an means not or without. So anaerobic cellular respiration happens without oxygen. That brings us to our next step. Our book calls this step pyruvate oxidation. Some places call it the prep reaction or preparatory reaction, but this happens in the mitochondrial matrix, which is the liquid portion of the mitochondria, and it requires those two pyruvates from glycolysis and NAD, a coenzyme, and it produces acetyl coenzyme A and also carbon dioxide and NADH. The NADH and acetyl coenzyme A is useful. The carbon dioxide is a waste product. So this picture is just showing the pyruvate, that 3-carbon molecule, making its way into the mitochondria for pyruvate oxidation, where the mitochondria produce NADH and acetyl coenzyme A. The next step is the citric acid cycle, which some people call the Krebs cycle. This also happens in the mitochondrial matrix, so the liquid part of the mitochondria, and this requires 2 acetyl coenzyme A, 6 NAD, and 2 FAD for coenzymes, and the final products would be 4 CO2, 2 ATP, and 6 NADH and 2 FADH2. So the citric acid cycle is a metabolic cycle with 8 steps, and each of those steps is catalyzed by a specific enzyme, and it's called the citric acid cycle because in the first step, the acetyl is removed from that acetyl coenzyme A and attached to oxaloacetate to form citrate, which is citric acid. And the next 7 steps, you're going to be breaking down that citrate and decomposing it back to oxaloacetate. That's what makes it a cycle. So NAD and FAD are going to pick up hydrogens and electrons, and then they'll later move those hydrogens and electrons to the electron transport chain. So don't memorize this picture, but this picture is showing you the citric acid cycle, and it's showing you pyruvate going through pyruvate oxidation, so you get acetyl coenzyme A. The acetyl coenzyme A going in, and the coenzyme A being kind of ripped off here and forming citrate. So that's how you get the citric acid cycle. It also shows you, like, along the way, there's different enzymes involved to do each step of the citric acid cycle, and it shows you the NAD and the FAD picking up hydrogens and electrons and becoming NADH and FADH2. It also shows the production of carbon dioxide there as well. So which 2 reactions in cellular respiration occur in the mitochondrial matrix? That would be the citric acid cycle and pyruvate oxidation. Number 9, how many ATP are produced in the citric acid cycle? That would be 2. So oxidative phosphorylation and chemiosmosis is our last reaction. Sometimes we just call this the electron transport chain. Both are good names. It's called oxidative phosphorylation and chemiosmosis because oxidation is happening by the electron transport chain because it's literally doing oxidation. It's transferring electrons. And then phosphorylation happens by ATP synthase. ATP synthase, it's a protein that moves protons across the membrane, and when those protons go across ATP synthase, that provides the energy required to make ATP. And then chemiosmosis, we remember what regular osmosis is, or just plain osmosis. That's movement of water across a semipermeable membrane. Chemiosmosis is movement of ions across a semipermeable membrane, in this case through ATP synthase, and that's used to make ATP. So this reaction happens in the cristae of the mitochondria, and if we're talking about aerobic cellular respiration, it requires 6 oxygen, and it also requires the reduced coenzymes NADH and FADH2. We got got those NADHs from glycolysis, pyruvate oxidation, and the citric acid cycle. And we got FADH2 from the citric acid cycle, which is also called the Krebs cycle. So this produces water and about 26 to 28 ATP per molecule of glucose. And then the NAD and FAD can be used later. So the electron transport chain uses electrons that originally were in your carbohydrates or your food, but then they got passed on to NAD and FAD, so we got NADH and FADH2. And the electron transport chain is literally a series of protein complexes in the inner membrane of the mitochondria. And so NADH and FADH2 donate hydrogens and electrons, and those hydrogens are pumped across this inner mitochondrial membrane, and then the electrons are passed down this electron transport chain. So we end up with a hydrogen ion concentration gradient, and we need to pass the electron to something. We can't just have free electrons like floating around. So oxygen in aerobic cellular respiration is the final electron acceptor. So that oxygen binds electrons and binds to hydrogen, so we get water. And the movement of these electrons creates this hydrogen ion or proton concentration gradient, and that provides energy for the next step, which is when we actually synthesize the ATP. So ATP synthase is this big kind of whirly protein complex in the inner mitochondrial membrane there. And these hydrogen ions or protons can't pass through the lipid layer here of the mitochondrial cristae, so they have to flow through ATP synthase. When they flow through ATP synthase, it actually spins ATP synthase, and that provides the energy to stick this phosphate group onto ADP and produce ATP. So we're going to watch this video together on biological gradients.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins.
So that's actually the electron transport chain and ATP synthase that they're showing you, and they're zoomed in on that mitochondrial cristae, that inner membrane in the mitochondria.
Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
So you can see those hydrogen ions are going through ATP synthase. It's turning ATP synthase kind of like a water wheel or a windmill, and that's providing the energy to make ATP. So to link ADP.
The process has now
and a phosphate
completed,
group.
and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP.
So this is kind of a simplified diagram of oxidative phosphorylation, or the electron transport chain, and chemiosmosis. So it's showing the NADH and the FADH2 dropping off their hydrogens and their electrons. Those electrons will be passed down the electron transport chain, and then oxygen is the final electron acceptor. That oxygen will accept electron, bind to hydrogens, and we get H2O or water. Then those hydrogen ions are set up in this hydrogen ion or proton concentration gradient, where there's more hydrogen ions on one side of the membrane than the other. So those hydrogen ions then can flow through ATP synthase, which gives the energy to make ATP from ADP and a phosphate group. So to big picture review, we talked about cellular respiration with glycolysis. Glucose goes into glycolysis. You also have to spend 2 ATP. You get a net gain of 2 ATP because you actually make 4, but you spent 2, so your take-home pay in ATP is 2. You get 2 pyruvate and you get NADH. That NADH will later be used in this electron transport chain. The pyruvate goes into the next step, pyruvate oxidation, and you get acetyl coenzyme A and NADH, but no ATP in this step. That acetyl coenzyme A goes into the citric acid cycle. You get 2 more ATP, and you get NADH and FADH2. All of those NADHs you made and FADHs are going to go into the last step, which happens in that inner mitochondrial membrane, the cristae. This step our book calls oxidative phosphorylation. A lot of times people call it the electron transport chain. And in this step, those hydrogens and electrons are ripped off of NADH and FADH2, and we build a proton concentration gradient. Those protons flow through ATP synthase, and we make a boat ton of ATP. So 26 or 28 ATP. So why can't I give you an exact specific ATP amount? Most of the energy flows in the following sequence, from the glucose to NADH to the electron transport chain to that proton motive force, where protons flow through ATP synthase and then ATP. About 34% of the energy in a glucose molecule is transferred to ATP, so you get about 32 ATP total, but different places are going to give you slightly different estimates. Lower estimates account for the transport of ADP into and ATP out of the mitochondria. Also, that proton motive force can be used to do other cellular work as well. Number 10, where is the electron transport chain located? That's on the inner mitochondrial membrane, the cristae. Number 11, which step of cellular respiration produces the most ATP? That is the electron transport chain, or the oxidative phosphorylation and chemiosmosis. Number 12, which step of cellular respiration produces the most carbon dioxide? That's going to be the citric acid cycle. And for number 13, which step of cellular respiration produces water? That's going to be our last step, the electron transport chain. So what if the cell doesn't have oxygen available to it? So it could either be in a low oxygen environment, or it could be a part of the organism that's deficient in oxygen or deficient in oxygen. So what could cause an oxygen deficit in an organism? If you were doing something like sprinting and your muscles were working as hard as they could and they were using up the oxygen to make ATP, then you could have an area that was oxygen poor in your body, even though you're breathing and even though, you're alive and doing well. You've just used up all of the oxygen locally available. So if cells lack oxygen, what they're going to do is either use anaerobic respiration or fermentation. So anaerobic cellular respiration uses something other than oxygen as the final electron acceptor in the electron transport chain. And fermentation doesn't do all the steps and only does substrate level phosphorylation. So anaerobic cellular respiration still involves glycolysis, the citric acid cycle, and the electron transport chain. The only difference really is is instead of oxygen as the final electron acceptor, it has a different electron acceptor. For example, E. coli uses nitrate under anaerobic conditions. Some organisms use sulfate. The exact electron acceptor is going to be species dependent, but if it's anaerobic, it would not be O2, oxygen gas. So fermentation, on the other hand, produces 2 ATP per glucose molecule when oxygen is not available. This is much less than cellular respiration, which yields about 32 ATP. So with fermentation, the pyruvate is reduced to either lactic acid or ethanol, depending on the organism that's doing fermentation. So this does provide a rapid burst of ATP when oxygen is not available, but the products like lactic acid or ethanol are toxic to cells, and it's a lower production of ATP. So these two pictures here are just showing fermentation in yeast. And so in this case, yeast that's making wine would produce ethanol, which is alcohol, and carbon dioxide. And then this person is hitting the gym hard and they have, an oxygen poor area in their bicep because they're really working those bicep curls. They're going to do fermentation for a quick burst of ATP. So they do glycolysis, but you don't get to keep the NADH. Instead, you pull those hydrogens off and you get lactate. So the exact outcome of fermentation is, again, species dependent. There's many different fermentation products. So fermentation is different from respiration. Both oxidize glucose to pyruvate in glycolysis with a net gain of 2 ATP, and NAD is reduced to NADH. But what happens to that NADH is going to depend on if they're doing fermentation or respiration. So in fermentation, the final electron acceptor is going to be an organic molecule like pyruvate or acetaldehyde. So you could get lactic acid or ethanol and carbon dioxide. Whereas in cellular respiration, the final electron acceptor is going to be, ultimately it'll be oxygen, but the NADH is going to transfer those electrons to carrier molecules in the electron transport chain and set up that proton motive force that ATP synthase uses to make ATP. So we absolutely eat more than just glucose. We just didn't go into every other organic molecule because it's hard enough to learn just this first part, right? So glycolysis accepts other carbohydrates besides just glucose. Proteins need to be broken down into amino acids before they can be kind of fed through glycolysis or the citric acid cycle. Fats are digested to glycerol and fatty acids can be broken down by beta oxidation and yield acetyl coenzyme A. And actually, an oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate. So the body uses these various small molecules to build substances as well. So we don't just use proteins to make ATP, we use proteins for their amino acids to build other proteins. So some of these small molecules come directly from food and others can be produced as we do glycolysis or the citric acid cycle. So let's review. Number 15 says in a disorder called pyruvate dehydrogenase complex disease, the enzyme responsible for converting pyruvate to acetyl coenzyme A is inhibited. Which of the following would you expect to happen? So this would probably be a good one to pause and think through before you just hear me spit out the answer to you. So if we said that the enzyme that goes from pyruvate to acetyl coenzyme A is inhibited, that means we're not going to get any acetyl coenzyme A, or we'll have less acetyl coenzyme A. And then what would happen with our pyruvate levels? If we can't go from pyruvate to acetyl coenzyme A, we should have higher pyruvate levels. So let's look at that first. Let's see if there's any we can eliminate. So the first one, pyruvate goes up. That's okay. Second one, it says pyruvate and lactate would go up. That would be okay. Pyruvate, acetyl coenzyme A, lactate all go up. And D says they all go down. So we can eliminate D just knowing that pyruvate is going to increase if we can't convert pyruvate to acetyl coenzyme A. So we got our pyruvate figured out. And then we said since that reaction is inhibited, acetyl coenzyme A levels are going to go down. So A says pyruvate goes up, acetyl coenzyme A goes down. That could work. B says pyruvate goes up, acetyl coenzyme A goes down. That could work. C says everything goes up, so C is a lie. So now what's tricky is we have to think about lactate, right? How do we get lactate? We get lactate when we are doing fermentation. When would we do fermentation? We would do fermentation when we can't do the rest of aerobic respiration. So the lactate levels would actually increase because we can't go on to the citric acid cycle if we don't have acetylcoenzyme A, so we'd have to do fermentation. So 15, the correct answer is B. Pyruvate and lactate levels would go up, acetylcoenzyme A levels would go down. Number 16, does fermentation require oxygen? No, it does not require oxygen. 17, is anaerobic respiration the same as fermentation? Explain. No, anaerobic respiration makes about 30 ATP, fermentation makes about 2 ATP. Anaerobic respiration goes through all of the steps of cellular respiration. It just uses a different final electron acceptor than O2 oxygen. Fermentation basically just gets stuck at glycolysis. So they are not the same. They are both anaerobic processes, meaning they don't require O2 oxygen, but they are not the same. So we made it to the end of chapter 7. Energy flows into most ecosystems as light, and then photosynthetic organisms use that light energy to produce a carbohydrate, and then photosynthetic organisms and heterotrophic organisms use cellular respiration and fermentation to produce ATP. We talked about oxidation and reduction. Oxidation is loss of electrons, reduction is gain of electrons, and we said for this class you can kind of shortcut it by tracking hydrogens because the hydrogen and the electron are generally transferred together. The coenzymes would be NAD and FAD in this chapter. When we get to photosynthesis, we'll talk about NADP. As far as ATP production in general goes, you can do substrate level phosphorylation or oxidative phosphorylation and chemiosmosis. Oxidative phosphorylation and chemiosmosis is where you have your electron transport chain and ATP synthase. So big picture cell respiration is for the production of ATP. Mitochondrial anatomy, the liquid part of the mitochondrial is the matrix, and the inner membrane is called the cristae. And then we went through the reactions of cellular respiration. So we had glycolysis, oxidation of pyruvate, the citric acid cycle, and the electron transport chain. We compared aerobic and anaerobic cellular respiration to each other. Anaerobic cellular respiration has a different electron acceptor than O2 oxygen. Aerobic cellular respiration, O2 oxygen, is the final electron acceptor in the electron transport chain. And then we said you eat more than just glucose and you use the products of your digestion, like your amino acids and things like that, for more than just energy. Like you're going to use amino acids to build additional proteins as well. So that is chapter 7.